Welcome back. In the previous session, we have found out that DNA is the genetic material in most of the organisms. In this session, we will be discussing in detail about the chemical structure of the deoxyribonucleic acid molecule, and also we will be discussing how is this DNA present or packed inside the nucleus. Let us begin with the structure of the DNA. DNA stands for deoxyribonucleic acid. So chemically, DNA is acidic in nature, and it is a nucleic acid. That is, this substance is present inside the nucleus. This is the same substance which Friedrich Miescher called it and named it as nuclein. He did not have any idea about the DNA, but he found that certain acidic substances were present inside the nucleus and he called them nuclein. Deoxyribonucleic acid is made up of three important components. If you look at the structure of DNA as such, it looks like a long chain of components that are sewed together. Therefore, we can say that the DNA is a polymer. It is made up of many repeating units. These repeating units are what are called as nucleotides. So the basic structural unit in a DNA is nothing but a nucleotide. So when many nucleotides they are joined to each other, it forms a polynucleotide chain. So DNA is nothing but a chain of polynucleotides. Let us look at the structure of the nucleotide, which is the basic structural unit. A nucleotide is composed of a nucleoside and a phosphate group. The nucleoside in turn is made up of a sugar and a nitrogenous base, about which you have already studied in your first PUC lesson, biomolecules. Nonetheless, we will quickly recollect about the structure of the nucleotide. Let us start with nucleoside, the sugar and the nitrogenous base. In DNA, the sugar is called as deoxyribose sugar, whereas if it is RNA, it is called as a ribose sugar. The structure of one such sugar molecule is drawn. So this sugar molecule, as you can see, it is a pentose sugar. So there are 5 carbon atoms in this sugar molecule, starting from this naming from here, 1 prime or 1 dash, 2 prime carbon, 3 prime carbon, 4 prime and 5 prime. So it is made up of 5 carbon atoms in a pentagonal structure, and the difference between a deoxyribose sugar and ribose sugar is that in a deoxyribose sugar there is 1 atom of oxygen missing at the 2 prime position. So in a DNA we have 2 prime H, whereas in a RNA we have 2 prime OH. So deoxy, 1 oxygen atom is missing at the 2 dash or 2 prime position. Hence we call it as a deoxyribose sugar, whereas this type of sugar is present in RNA molecule. Attached to the pentose sugar, we have the nitrogenous base, or sometimes simply called as a base. This nitrogenous base is of two types. There are purines and there are pyrimidines. Purines are double ringed structures, whereas pyrimidines are single ringed structures which are cyclic in nature. There are two types of purines, adenine and guanine, which are made up of two rings, whereas pyrimidines are cytosine, thymine and uracil. In DNA, we find only cytosine and thymine as pyrimidines, whereas in RNA, instead of thymine, we have uracil in its place. Chemically speaking, thymine is nothing but 5-methyl uracil. So RNA contains uracil instead of thymine. So these are the nitrogenous bases. The nitrogenous bases are attached to the ribose sugar or deoxyribose sugar using a glycosidic linkage. So here we have drawn one such nitrogenous base. This is our adenine, double ringed purine structure. This nitrogenous base is attached to the first carbon of sugar. So this is a condensation reaction, and this is attached to the nitrogen. So this linkage is what is called as glycosidic linkage or a glycosidic bond, by which a nitrogenous base is attached to a sugar molecule, glycosidic linkage. This unit, a nitrogenous base plus pentose sugar, is called as a nucleoside. In this case, because the sugar is deoxyribose, we can call it as a deoxynucleoside. To this nucleoside, when the phosphate group is attached, the acidic nature of the DNA is due to the phosphate group. So where does the phosphate group attach to the nucleoside? The phosphate group attaches to this particular fifth carbon molecule, the 5' carbon, so this is our phosphate group that is attached, and this linkage is called as the Ester linkage. The bond between a phosphate group and a sugar molecule is called as a Ester linkage. Thus, this is our nucleotide called as deoxyadenine, which is the nucleotide. Now, like this is one nucleotide. Many nucleotides are connected end to end to form a long nucleotide chain. So hence we call DNA as a polynucleotide chain. So let us see how is one nucleotide connected to another nucleotide. So here we have two sugar molecules, and let us say we have one more. So this is the base, a nitrogenous base. It could be any base. And how is this nucleotide connected to this nucleotide? What happens is a phosphodiester bond is formed due to condensation reaction. So the reaction is taking place between the 5 carbon of one nucleotide and the 3 carbon of another nucleotide. So the 3 prime end here is open and the 5 prime end here is open. So this OH molecule is lost and the hydrogen molecule is lost due to condensation, and water molecule is removed. When the water molecule is removed, a phosphodiester bond is produced. So this bond is what is called as a phosphodiester bond. So two nucleotides are connected to each other by a phosphodiester bond. It is called a diester because two hydroxyl groups of two molecules are reacting with each other. This is what is called as a dinucleotide because there are two nucleotides connected. Similarly, this keeps on joining to other nucleotides forming a long chain. That is how the DNA looks like. Erwin Chargaff, these are now famously called as Chargaff's rules. According to Erwin Chargaff, the total percentage of purines in a DNA molecule is equal to the percentage of pyrimidines, and also the percentage of adenine is always equal to the percentage of thymine, and guanines are always equal to that of cytosines. These are the observations made by Erwin Chargaff. Purines is always equal to pyrimidines, and the amount of adenine is always equal to thymine, and guanine is always equal to cytosine. So if you have to put it in the form of a formula, we can say that A plus T and divided by G plus C is not equal to 1. This is a unique ratio which varies from one organism to another organism. Every organism has a unique ratio of purines and pyrimidines, and this can be used to identify and classify the organisms based on the percentage of purines and pyrimidines present. So A plus T divided by G plus C is not equal to 1. This value varies from one organism to another organism. This Erwin Chargaff's principle were used by James Watson and FHC Crick to propose the structure of the DNA. The famous DNA double helix model was proposed by Watson and Crick, called as the double helix model. What we have discussed till now is the structure of the nucleotide and how is one nucleotide connected to another nucleotide. Let us now look at the structure of the DNA in general. The double helix model was proposed by Watson and Crick. To explain the structure of a DNA polynucleotide. According to the double helix model, DNA is made up of two polynucleotide strands which are in anti-parallel direction. So you can imagine the DNA as a ladder. So this is one strand of a ladder, this is another strand of a ladder. This is, let us say, the backbone of the DNA, and this backbone of the DNA is made up of sugar plus phosphate, and the nitrogenous bases, they are present perpendicularly inside the molecule. So the bases are present perpendicularly inside the molecule. Now, in anti-parallel direction, this means that every DNA strand has a direction. This direction is called as the 5' and 3' direction. For instance, if you see here, two nucleotides are joined to each other. You can see there is a free 3' OH group here, and there is a free 5' phosphate group here. So we can say that this particular strand is in the 5' 3' direction, or 5' 3' direction. Because there are two strands in anti-parallel direction, if one strand is in 5' 3', the other strand is anti-parallel, that is in the opposite direction. This is what is meant by the two DNA strands are present in anti-parallel direction, with the sugar and the phosphate as the backbone. So if you have to draw this like this, so this is the sugar molecule and the phosphate molecule that is attached to it, the 5' direction free, and the nitrogenous base is present perpendicularly inside the molecule. Like this, you see that many nucleotides, they are connected to each other. So this is our phosphodiester linkage which connects the two nucleotides, and it is not a straight molecule but it is twisted. So if you take this ladder and twist it, you get what is called as a helical structure. So when you twist this ladder, you end up with the helical structure like this. So it is twisted in a right hand fashion. So we can say that the DNA molecule is made up of two helical polynucleotide strands which are twisted upon each other in a right handed manner. So it is twisted like this. The Hermite-Chargaff's rules say that the amount of purine is equal to amount of pyrimidine. In the base pairing, one strand of DNA is connected to another strand with the help of base pairs. Base pairing is done with the help of hydrogen bonds. So the two strands are linked by hydrogen bonds. The base pairing is always between a purine and a pyrimidine. So a purine always forms bonds with the pyrimidine. So let us say the purine here is adenine. On the opposite strand we find thymine. So adenine will always bond with thymine using a double hydrogen bond, and if it is guanine, it will bond with cytosine using a triple hydrogen bond. So adenine requires 2 hydrogen bonds and guanine requires 3 hydrogen bonds. Thus the 2 polynucleotide strands are joined. This is what is called as a base pair. The number of base pairs in every organism is unique. By counting the number of base pairs, we can, which is very unique to the organism, we can identify the organism. For instance, in human beings, we have a total of 6.6 into 10 to the power 9 base pairs in a human cell. That is approximately 6.6 billion base pairs. 10 to the power 9 means 1 billion. So this is in a diploid cell. If it is a haploid cell contains half of this, that is 3.3 into 10 to the power 9 base pairs of DNA is present. Similarly, in E. coli bacteria, we have 4 into 10 to the power 6 base pairs in E. coli. This number is unique to every organism. No, so base pairing is done between a purine and a pyrimidine. Because of this, the distance between the 2 strands is equal throughout the DNA molecule, because a double ringed purine is bonded to a single ringed pyrimidine. Everywhere the distance is uniform. The distance is found to be 2 nanometer between one strand and another strand. This distance is uniformly maintained throughout the DNA molecule. So the width of the DNA molecule is 2 nanometer. Because the DNA is twisted, we can find that the DNA can be divided into turns. So this is one complete turn called as its pitch. So pitch of the DNA is one complete turn. It is observed that in one pitch there are approximately 10 base pairs. 2, 3, 4, 5, 6, 7, 8, 9 and 10. So this one helical turn is called as the pitch of the DNA, which approximately contains 10 base pairs.
One pitch is equal to 10 base pairs, has an approximate length of 3.4 nanometer, as you can see in the diagram. So 10 base pair is equal to 3.4 nanometer. From here we can calculate the distance between the 2 base pairs present one above the other. As you can see in the diagram, the distance would be roughly 0.34 nanometer. Distance between one base pair and another base pair present in a DNA molecule is 0.34 nanometer.
Not only that, another interesting feature of the DNA molecule is that one base pair is not stacked upon directly over the other in a same plane. So the base pairs are not stacked one above the other like in a ladder. Instead, the base pairs are present at a particular angle. So if there is one base pair present here, the second base pair is present at an angle of 36 degree. So there is a twist of 36 degree that is present in between the 2 base pairs. So by the time one turn is completed, you can see 36 into 10, 360 degree, one turn is completed. So one base pair is stacked upon another base pair at an angle of 36 degrees. So this will keep on changing. So like this, and when one turn is completed, 360 degrees completed, giving rise to another pitch. So because of this, what we see is that there are small grooves, gaps that are formed. So this is what is called as the minor groove, and this larger gap what is seen is the major groove. So because of the helical twist, we can base pairs are present not in the same plane but at an angle of 36 degree over each other. That is another point in the structure of DNA. That is what the structure of DNA looks like, and you already know that the DNA is structurally and chemically very stable molecule. The structural stability of DNA is offered because of the presence of two strands, and the hydrogen bonds, they offer structural stability to the DNA molecule. Other than the hydrogen bond, we also have other small intermolecular forces such as the van der Waals force of attraction, and the fact that each base pair is stacked upon the other not in the same plane but at an angle of 36 degree, it offers extra structural stability, making DNA a very stable molecule. Now, once this structure of DNA was proposed by James Watson and Francis Crick, part of the article that was published was also influenced by the X-ray diffraction images that was given by Maurice Wilkins and Rosalind Franklin. The X-ray diffraction images were used by Watson and Crick to propose the double helical model of DNA. So to quickly summarize, DNA is made up of two polynucleotide strands which are in opposite direction, anti-parallel, 5' to 3' and 5' to 3'. The backbone of the DNA is made up of sugar and phosphate. The nitrogenous bases are present perpendicularly inside the DNA molecule. The two bases on opposite strands are joined by hydrogen bond. Adenine will always pair with thymine using a double hydrogen bond, and guanine will pair with the cytosine. A purine is always bound with the pyrimidine, due to which the distance of 2 nanometer is maintained throughout the DNA molecule. In a single turn called as the pitch, there are approximately 10 base pairs, which is 3.4 nanometer in length. Therefore, the distance between the two base pairs is 0.34 nanometer. Into 10 bases will give us one pitch, and the two base pairs are present one above the other at an angle of 36 degree, not in the same plane, not one above the other. Also, by Erwin Chargaff's research, we know that the percentage of purine is always equal to the percentage of pyrimidine, and that A plus T divided by G plus C is not equal to 1. This is a unique ratio, very unique to organisms. In human beings, the length of the DNA molecule is 6.6 into 10 to the power 9 base pairs. That is the length of the DNA molecule. Based on the pairing of the nucleotides, Crick, that is Francis Crick, he proposed the central dogma in molecular biology. The central dogma, very simply, it states that the flow of information is always from DNA molecule, because DNA is the genetic material. The properties present in the DNA, it is transferred into RNA, and the information present in the RNA is transferred into proteins. This conversion of DNA to RNA is what is called as transcription, and the process of conversion of RNA into protein is called as translation. This is the direction of movement of information. All organisms pass information from DNA to RNA and from RNA to protein. Exceptional case is the virus, because in most of the viruses we have RNA as the genetic material. So in these viruses, especially the retroviruses, their genetic material is RNA. So they first convert the RNA into DNA by a process called as reverse transcription. And once again, DNA is converted into RNA and RNA is converted into protein. This is the central dogma of molecular biology. This was influenced by the double helix model, especially due to the base pairing nature of the DNA. Another very important point hidden in the structure of DNA is that, let us say this is one strand of DNA. If you know one strand of the DNA molecule, using the complementary base pairing principle, we can tell the second strand also, because strands are anti-parallel. The second strand would be 3' to 5', and the second strand, in the place of adenine we will have thymine, in the place of thymine we have adenine, in the place of guanine we have cytosine, guanine, CG, A. So if you know one strand of DNA molecule, you can find out the second strand of DNA molecule. This complementary base pairing is also useful during DNA replication, that is during cell division, during the S phase of the cell cycle, when DNA doubles itself, it copies itself in a process called as DNA replication. DNA produces DNA by DNA replication. The two strands, they separate, and both the strands will act as parent strands, which in turn produce the two daughter strands. The two daughter strands will have the exact information present in the original parent strand due to the complementary base pairing that is seen in the DNA molecule. This in fact was mentioned by Watson and Crick in the original article that they used to propose the model of double helix structure of DNA, wherein they mentioned that it has not escaped our notice that this complementary base pairing can also be used to explain how the information present in the parent is transferred to the daughter without much mistakes, how it is copied in a perfect manner owing to the complementary base pairing. That is all about the structure of deoxyribonucleic acid. The difference between DNA and RNA lies in the fact that RNA, in its 2' position, it has got one extra oxygen atom, it is ribose sugar. Instead of thymine, RNA contains uracil, and DNA has two strands, it is double stranded, whereas RNA contains only single strand. In the next session, let us discuss about how this long molecule of DNA is compressed inside a small area within the nucleus of a cell. Thank you.